Ô, Bruno, já tá no streaming, mas a gente não tá ouvindo aí direito vocês, não.
Tá. Não, isso aí, vamos começar agora. Isso aí, Lucas Bolini vai fazer a apresentação aqui.
Ah, tá, beleza.
Okay, so good afternoon, everyone, and welcome to another edition of our Immuno webinars. Today we have the honor and the pleasure of having Dr. Adriana Bonomo presenting to us. She graduated as a medical doctor here at the Federal University of Rio de Janeiro. She did her master's and and got her doctorate degree here in our institute under her doctorate under the supervision of Professor Marcelo Barsinski, who we also have the pleasure of having here today. She did a postdoc period in the National Institute of Health in the United States. She was a professor here at our university for almost 20 years, after which she moved to Oswaldo Cruz Foundation. And for the past decade, she has been a specialist researcher, the chief of the Themic Research Laboratory. She was also the vice president of the Brazilian Immunology Society. She's the coordinator of the Global Immunology and Immune Sequencing for Epidemic Response, or GII-SCR, under the Bill and Melinda Gates Foundation. And she's also the coordinator of the translational programs FIOCRUZ and FIOMUCOSA in the Oswaldo Cruz Foundation. So thank you very much for accepting our invitation, and the floor is yours.
Good afternoon. Thank you for the invitation. It's really a super pleasure for me to talk here, since my whole education in immunology and a very strong part of my formation was in this house under the supervision of this wonderful scientist that is sitting right in front of me. That really honors me to have him here. So I'm going to tell a story that we started like around 10 years ago, maybe a little bit more. And we are trying to get to a different point now. So some data are published, some data are not published, and the ideas, recently we published part of the ideas. So, and I'll have the thing here. So, the whole thing started when I was at INCA and I had to work with tumor. And then actually I found out something that everybody knows, that the dream of every tumor, whoops, wow, this goes big. The dream of every tumor is to colonize the whole body. This is the dream of a tumor. It's the end point that the tumor wants. But, and also, like primary tumors, usually we can treat on surgery and radiation, but metastases are, I'm having problems with this then, but metastases usually are not responsive to the regular therapies. Now with immune checkpoints, we have a little bit, I mean, some better results, but in general, metastasis is like an endpoint that is not treatable. So you just comfort the patient. And we know from this paper from 1889 that metastases are not by chance. So you don't have a primary tumor. This primary tumor sends or have like the escapees from the primary tumor that will go through the whole body. and they will install themselves and colonize different organs. It's not exactly like that. And what this guy showed, Paget, he autopsied or necropsied 700 and something patients that have died with cancer. And he looked at the metastases from those patients. And he realized that different primary tumors colonize different metastatic organs. So have different target organs for metastases. And so he came up with the hypothesis of the seed and soil, where the seed is the tumor and the soil is the tissue. And of course, for a seed to be able to thrive and grow and develop, needs a good soil. So that means that the tumor cell arrives to a certain tissue, and that tissue needs to be receptive or hospitable for that tumor. If it's not going to colonize. And with this, we can understand like these different patterns of metastasis from different primary tumors. So we wanted to work. We end up working with breast cancer for these clinical and epidemiologic reasons that are listed there. But the real reason was we wanted to have a model where we could have bone metastasis, because the idea was to work on the immune response inside the bone. And this because, so the reason we started turned out to be totally different from what exactly has happened. But the idea initially was bones are like good places to keep cells on a quiescent state. And we know, like in breast cancer, that the patient has its primary tumor taken out, go to radiotherapy and everything, and then like 20 years, 15 years later, the patient reopens like the disease with bone metastasis, or starting with the bone metastasis to the rest of the organism. So the idea was to do like osteoimmunology of cancer and metastasis. But at the end, this is not what I'm going to show you. So it started with a model. And so the rational reason is that breast cancer is actually in AL is the most frequent cancer, even if you add men and women. And we have like still today, 60% overall survival with 70,000 new cases per year in Brazil. And from those patients that have invasive tumors, around 70% or 75% will develop bone metastasis. So if the tissue needs to be hospitable for the cell that is coming in, one important thing for us to know is what is the basic homeostasis of this tissue, and how this basic homeostasis could generate a hospitable environment for the cell. So, and on the bone, it's sort of easy. It's super simplified what I'm showing here, but the bone remodeling system counts on osteoclasts. Osteoclasts are polykaryons of macrophage-like cells. They have special enzymes that are able to digest the mineralized matrix. And when they eat the matrix, when they digest the matrix, they release growth factors that are attached to the matrix. These growth factors will act on progenitor cells that I wrote here. Osteoblast progenitor could be a mesenchymal progenitor, and it stimulates the cells to differentiate into an osteoblast, and then the osteoblast adds the bone back in place. And so this is the cycle. And in 2002, Gregory Mundy, who unfortunately is already deceased, he came up with this vicious cycle hypothesis for bone colonizations. And I believe this vicious cycle will happen in every organ. It's not something that is specific to the bone. And where he showed, or he gathered information where he could put up the following hypothesis. When the osteoclast, digests the matrix, it releases growth factors. Those growth factors and differentiation factors are actually factors that will feed the tumors. So will work as a simulator for the tumor to grow. And then the tumor produces substances that will hyperactivate the osteoblasts. And then the osteoblasts, when they are hyperactivated, they produce tons of this molecule called RUNC ligand. We're going to talk a lot about it. And this molecule is the factor for the differentiation of monocytes into osteoclasts. So with this, he said, maybe a little bit before, we know that sometimes a lady will go into an X-ray examination and her bone will be full of hypodense areas, indicating that this person has an osteolytic disease. This osteolytic disease was believed to be a consequence of the tumor colonization of the bone, where this cycle would be hyperstimulated, okay? Another thing that is important for us to consider here is, at one hand, we have this RUNC ligand that simulate the RUNC is the molecule that binds the RUNC ligand on the surface of a monocytic lineage, or even macrophage, or even dendritic cells. This same osteoblast can produce osteoprotegerine, it's OPG. OPG is a decoy receptor for the RUNC ligand. So when OPG is produced, it binds RUNC and avoids the ligation of the RUNC ligand into with the RUNC on the surface of the cell. So this is the establishment of bone colonization will depend, or will interact very vividly with the remodeling system of the bone. And the bone is very active. I mean, we redo our bones at every, let's say, 20 years. We have we have whole new bones we renew it at every 20 years, the whole thing. So it's super active. Besides other things that happen in the bone, like the hematopoiesis, control of calcium, and et cetera, et cetera. So then our question was if there was any interplay between the mammary tumor and the bone, and if this could be mediated by the immune system, okay? And at first, if you're thinking about seed and soil, it's really weird, like, a mammary gland is a mammary gland. It's going to make milk, right? And the bone is involved in hematopoiesis, is involved in calcium control, is involved on keeping the bone healthy and sort of new with this renewal process. So it's like weird, but let's see what's going to happen. So Ana Carolina Monteiro, who worked with me for many, many years, she's also an egress from this institution. Now she's a professor at the Fluminense Federal University. She's still working with the same model. So Carol could establish this 41 model. That's a very interesting model on my view, because we have sibling cell lines that were originated from the same primary tumors, but have different metastatic capacities. So we have this 41. That's like a triple negative in women. It's one of the most aggressive forms of breast cancer, and especially there's no specific treatment for it. There's no target molecule or anything like that. And it's very metastatic. It goes to lymph nodes, spleen, bone, lung, liver, and central nervous system. And it has a sibling that's called 67, and there is an in situ tumor. So they were all originated from the same primary ones, but they have different metastatic capacities. The other good thing about this model is that they come from a biopsy animal that's immunocompetent. So if you want to study the immune system, especially the adaptive immune system, I need to have the whole animal working. And this is a curve of the metastasis occurring in the lymph nodes or on the bone. Okay, if we look on this assay that we called clonogenic metastatic assay, it's an assay where we will look at the cells that can survive and will grow in vitro. So take out the organ and culture, they have a resistant gene for one drug, so we can select the cells that are going to grow. And if we look at, and this information will be important for us from now on, like on the lymph node before, so metastasis will appear after day 11 and before day 14. And this, we also did a PCR for cytokeratin 19 that's not present in the bone marrow or in the lymph nodes. And we see here that on the lymph node on day 12, we don't have metastasis, or we don't have any material from the primary tumor in this organ. And it appears after day 14. So it's if you want to work before metastasis with lymph nodes, has to be before day 12, and on the bone marrow only on day 16. And here is after day 18. So just keep this in mind. Okay, then so add the tumor to the biopsy. We inject on the 4th mammary pad, and then we count the number of osteoclasts that these guys have on their bones. So this is a naive animal. This is the guy that receives the non-metastatic cell line, and this is the guy that receives the metastatic cell line. And we see that there is an important increase on the number of osteoclasts on that bone. It's counted like this red thing here. Is a special staining to identify the cells. And when we look at the bone mass, look how interesting. In 6 days, the guys already lost like close to 50% of the trabecular mass of the bone. And by this time, there is no metastasis yet. So this is an osteolytic disease that was not induced by metastasis, and it's very early. So then let's see what the T cells that are present in the bone marrow are producing in terms of cytokines. And so the darker line is the 41 and the light line is the 67. And what we can see here is that T cells that come from 41 produce by day 11 already a good amount of IL-17, of RUNC ligand when we compare to the 67, IL-1 beta, and IL-6. And those cytokines, all of them are pro-osteoclastogenic cytokines, are cytokines that induce osteoclastogenesis and activation of this osteoclast. Down here, it's an in vitro assay just to check if the supernatant that we could get from these T cells after they were stimulated, if they could induce osteoclastogenesis in vitro. And so this is the control. So to induce osteoclast, you need RUNC ligand and MCSF to expose the RUNC, and RUNC ligand to induce a differentiation. With the supernatant from naive animals, we have nothing. And this is soluble antigen, because we use soluble antigen to simulate the T cells. And when we add like the supernatant from T cells that come from the guy that received the 41, and we simulate them in vitro, we increase the number of osteoclasts that is being produced. This is inhibited by OPG as an antagonist for RUNC ligand. But there was another molecule that was on the scene by then. It was IL-17, because IL-17 is involved in arthritis, for example. So it's involved in some osteo diseases, bone diseases, especially inflammatory diseases. Then this is not the case here. But just like the antibody to IL-17 F especially didn't work at all. So apparently it's mediated by something that the T cell can do that is inhibited by OPG, so probably it's RUNC ligand. This is number of cells, this is activity of TRAP, it's the enzyme that is specific for this cell. And we can also do resorption area. If you do the culture of the cells on top of a matrix that mimics the bone matrix. And you can see, and the osteoclasts, after they are formed, they sort of bite that thing. And then you stain it with silver, do a silver stain, and you see the holes on the, and then you can measure the area that is absorbed. So when you have osteoclasts, you have a high absorption area here and here. If you add the supernatant from the T cells, you also have a good absorption area, and it's inhibited by OPG. So much for this. So then the next question is, if the guys don't have, they have, much before they have metastasis, they lose bone mass. The T cells that are inside the bone marrow can produce osteoclastogenic cytokines. Let us transfer T cells from this bone marrow before metastasis is there. So we usually do by around day 11. Transfer it to a nude that doesn't have the T cells, stimulate with antigen, and look at the bones. So, and to check if the T cells by themselves, in the absence of tumor, if they can induce bone loss. So this is the bone mass of the nude that receive only the antigen, the nude that receives T cell from a naive animal, the nude that receives T cell from an animal that received a tumor that is not metastatic, and if they receive the T cells that come from the guy that received the metastatic cell line, the 41 plus antigen, we have bone loss. This is measured by histomorphometry, and this is measured by CT scan. So then we did a lot of other things that I'm not going to show the details, but now the question was, is this thing really mediated by RUNC ligand that is present on the surface or that is produced by the T cells? So how did Carol did that? It's the exact same experience, the experiment. The difference is that the T cell that we get from this guy that got the tumor, got the T cells from day 11, and then we silence with shRNA and transfer the silenced cells to the nude. I'm not showing all the controls. It was a very hard protocol to get working, but we managed to get it working, inhibiting when you use the SH for RUNC ligand, only the RUNC ligand, not inhibiting the IL-17 and vice versa. So it was okay. We spent one whole kit just to do the settings. And so this is the bone of the guy that receives the T cell from the 41. This experiment is exceptionally good. If we silence the IL-17, nothing happens. It's still losing bone. If we silence the RUNC ligand, it doesn't lose bone. It's just like the controls here, okay? This is the double silence because we thought that probably, but we were wrong. And we would need IL-17 and RUNC ligand for that, but only the RUNC ligand is enough. So we take out, so the thing is like this, I take out the RUNC ligand from T cells only, and then I get no bone loss, right? Then, if Gregory Mondy was right, and if I inhibit the osteolytic disease beforehand, so I add the tumor, and then I have a situation where this situation here, oops, sorry. Then I have this situation here. So I have this situation, and if I have this situation here, I don't have the osteolytic disease. If the osteolytic disease is important for the tumor to colonize, the T cells would be working on the pre-metastatic niche and preparing the bone to receive the tumor. And then when we look at the metastasis, first thing that was really impressive is that the nudes almost do not make any metastasis. If we give to the nude the T cell that comes from the 41 guy, it does have the metastasis. This is the 41 silenced with the scramble. And if I silence with broncholigand, I have no metastasis at all. So we believe that we are adding one extra step for the vicious cycle. And this extra step would be a preparation of the metastatic niche. So the pre-metastatic niche setting that is dependent, and it's done completely by the T cell alone. And it has to be a T cell that produces this RUNC ligand. Just as a curiosity, RUNC ligand is the master regulator of bone remodeling, but it was first described on T cells. And it was, when it was described in T cells, it was had not by that time been described in the bone yet. And then the whole story of the RUNC ligand on T cells is that it would be important, but the data is not like that data that you have a big, a big thing and then a little thing. But apparently it's involved in memory, but it's also involved in tolerance. I'm not going to talk about this part, but just as a curiosity, since we have a lot of students here, and it's also important, like for the expression of air in the thymus for inducing tolerance on the thymic epithelial cells. It's dependent on RUNC ligand also. It's also important for the formation of the lymph nodes. So in the absence of RUNC ligand, you don't have. So I'm telling this, I'm talking about this thing because we have this idea that some molecules, or especially molecules of the immune system, the molecule belongs to the immune system. If you don't have it, this molecule. And I mean, each day more, we've been seeing that the same molecule can be used on different organs for different things, and then even in ontogeny on a different way from it's used on the adult, like tau. Tau receptor is great. It was described on an ontogeny experiment. It was not supposed to go to the immune system, but it did, because it's an economy. I think it's an economy basis of biology that our body uses. So, and the most important thing is that RUNC ligand is the important molecule for breast development during lactation. So breast cancer cells, many of them have RUNC ligand. Ours have, but when we do that, we delay the growth of the primary tumor. So, and we have metastases to other organs, but we don't have to the bone. So it mediates breast cell cancer growth, it does, but it has, when it is on the T cells, it has an important feature on the bone itself or inside the bone itself. Okay, so we believe that thing. And then we ended up going, let me see, because I talked too much. Okay. We noticed actually that when we took out the bones from the animals, the bones injected with the 41 tumor, the bone is very fragile. It's not very hard to macerate. We treat with enzymes and everything, but it's not very hard to macerate. It's a sort of soft bone. And the bone from the guy that received this non-metastatic cell line was super hard and very hard to macerate. And I had shown, so when we look at the bone mass, I think I have a thing. So the bone mass of the 67. And again, we did that a little bit before also, but again, it's very fast, the gain of bone mass with the 67. When we look at the osteoclasts that I have already shown this data, at the osteoclasts around the bone, they have much less osteoclasts. So when we look at the tissue, so Carl started looking at different cells and see what was happening. So this is the graph representation, but I'm going to show it here because I think it's cutest. And we can see better, right, than the line graph. So what we are showing here, what we are showing here is, this is a naive bone. We have the osteocytes inside the bone. Here, here, here. The guy that received the 67 has more osteocytes, first thing. Second thing, osteoblasts. Osteoblasts, they can be like this, very thin, and they can be like this or like this. When they are activated, they sort of get fat and turn up to be cuboid. And we have this cuboid cell. So look at the difference at this, with a lot of matrix that's not mineralized yet, and the regular bone, and the bone from the, and the bone from the 47, from the 41 guy. Okay, so we have more cuboid cells, and we even have more bone lining cells, and we have more osteocytes on the 67. So then what's happening? We know that this guy doesn't produce rank ligand, all the things with rank ligand, for this guy is negative. And there was this one that up to now, it's the only one paper that I know involving T cell and bone mass achievement on a regular basis, physiologically. So this paper showed that so this is the bone, and inside here we see the trabecular. So the animals that don't have B cells, they don't have, they have a very fragile bone with very little trabecular mass, okay? And they also produce very little OPG. So when we look at the cells that could be producing OPG, because until then, the big producers of OPG were believed to be the osteoblasts on the regulation for the rank ligand osteoblasts, rank ligand OPG. But they showed that if you don't have on the bone marrow, if you don't have B cells, you have low OPG production. And if you replenish the guys with the B cells, the OPG production goes up again. And when we look at which type of B cells would be producing these things, they are the mature B cells that are inside of the bone marrow. I don't know what is the teleological logic of this thing, but I believe I would think on dislodging hematopoietic niches to make space for the B cells to be produced. It's a regulation of what we like to call like floating niches that go to one side and to the other of the bone marrow. But this is another subject. So then we went up to look at the B cells. Okay. So this is bone. So this from now on, every red is 41, every blue is 67, every white is the control. Okay. So this is production of OPG, whole bone marrow. Lymph nodes do not produce OPG. When you isolate the B cells from the bone marrow, of course, only the B cells that come from the guy that received the 67 non-metastatic tumor produce OPG. So this was our control on the rank ligand. And so Ara said to me, I'm going to do this experiment. And I said, don't do it. It's not going to work. It's not going to work. Of course not. We're going to transfer B cells that are coming from a guy that received the tumor, blah, 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 to a totally full animal. So the B cells, they're not going to stick. We would need to have a mean count to do this. And she said, I have everything I'm going to do. So I'm glad that she did. And this is always a good thing to say when we have students. If you trust yourself, go ahead. So then we transfer the B cells from the bone marrow to a biopsy animal and gave the 41 tumor to this guy and check what kind of interference in terms of bone mass and metastasis this B cell will do. We have part of this published. I should have put it there, but I forgot. So when we look at the bone, so this is the control, like the control biopsy, the biopsy that received a tumor, the 41, and that received the 41 and naive B cells. Okay, so this is what we expected. I would expect this for the whole thing, but then yeah, if the B cells come from the guy that was injected with the 67 tumor, we block the bone loss. So it comes from here or here to here. Okay, and then she did combinations with T cells that didn't matter at all, especially because now we know that the OPG production is independent of the T cell. I mean, to be simulated at first, it depends on T cell because the node doesn't produce OPG, but after it's simulated, it doesn't need a T cell anymore on the second simulation. Okay. And when we look at the metastasis, again, we have what we, oops, I'm having, okay. So we have metastasis on the guy that received the tumor. If that same guy gets T cell from a naive animal, it still have the metastasis. If the B cells come from the 67, you block metastasis completely to the bone. What makes sense if this is dependent on OPG. So then Carol silenced the B cells with SH for OPG. And so when we block the OPG that are coming from the purified B cells, we do not have we still have a bunch of osteoclasts here, just like the animal that received only the tumor. But if the animal received the B cell that comes from the 67, we don't have osteoclasts. And when we look at the bone, we see what we would expected. Low bone for 41 plus B cells from 67 is high. And then if we take out the OPG from this cell, we again will have bone loss. And for the metastasis is the exact same thing that we saw or that we would expect. So it's the same thing here and here. And I'm going to show just the bone for sake of time. This is the amount of metastasis we have when the animal received the tumor. If the animal received the tumor plus B cells from the 67, okay, from B cells from the 67, we have no tumor, no metastasis. If we block OPG, metastasis kick in again. So it's apparently what we have here is a crosstalk between T cells and B cells, where one can inhibit the other, producing different things. And the funny thing is that this is simulated by the primary tumor up there on the very beginning of the disease. And we wanted to move to humans. We didn't finish yet, but I'm going to show the preliminaries. And what we did first, because in humans, we only have access to primary tumor or lymph nodes, right? Or blood. I don't like blood, but primary tumors and lymph nodes. Blood, you're laughing. Blood is the road, right? I used to say that you want to know what's happening in Rio and São Paulo, you study Via Dutra. It's something like that. I know that sometimes the only thing we have is blood, and we have to work with blood. Not that it's not informative, but it's very restrictive. So here what we did was let's look at this thing here because it's easy for me. It's easier to see. It's the same thing. So we purified by FACS sorting T cells and B cells from primary tumor and lymph nodes. Okay, so reds are 41s. Okay, so these are CD4 T cells purified, and this is quantitative PCR for rank ligand. And this is in the primary tumor and in the lymph node, or maybe so the other way around. No, that's it. Primary tumor is the square and lymph node the blue. Okay, and the B cells do not produce this thing. When you get we look at the OPG, only the B cells from the primary tumor produce OPG, and the lymph node cells, as we had seen on the ELISAs that I showed, remember the lymph nodes produce nothing, and then the B cells from the bone marrow produce OPG. So the lymph nodes do not produce, meaning for us that when we go to humans, we will need to use the primary tumor. But also it brings another, I think, important information, is that the tumor is modulating everything apparently inside the tumor itself. Maybe doesn't need to go to the lymph node to get everything activated. It's another dogma that we need to work with. And this is it's very preliminary. We already have maybe 3 more. So the idea was to have a proof of principle with 10 patients looking at primary tumor, from 10 patients that have bone metastasis later, and 10 patients that on the same time frame did not have bone metastasis. And we looked at CD3 rank ligand positive cells on the primary tumor, on the guys that made bone metastasis, and the guys that do not. We did look at the OPG, sort of a mirror thing. When we do the Carol, I love ratios. So let's do the ratio, because the ratio is going to be much more. I mean, you add results, and so we have maybe something that will be more clear to us. And this is how we are now. So if you do the ratio for CD3 rank ligand, CD20 OPG, guys that have bone metastasis have a high ratio compared to the guys that do not have bone metastasis. So this could be really good, because we can predict. The idea is that we can predict who is the patient that is going to have bone metastasis. And although there are many studies using anti-rank ligand antibody or biphosphonates that inhibit osteoclasts, things that will inhibit osteoclasts, and these things have no impact on bone metastasis. It's amazing. So, I mean, for us to move after we finish this, it's being made by a undergraduate student, but after we finish this, and then the idea is to build like a BiTE, a molecule that has an antibody with 2 binding sites, CD3 and rank ligand, to check if we can inhibit that. If with this too, we could inhibit bone metastasis for sure. So then, how much do I have? I have 10 minutes, right? Okay. So this is the message for us, okay? And on this system thing and tissue thing, there was a paper that came out a little bit before. We were already working on that. We already had our preliminary results, and this paper came out showing that on a different model of breast cancer, this YMT is a transgenic model. It's a B6, ours is a BALB-C. That for the YMT cells to colonize the lung, they only look at the lung. We look at everything. I didn't show, but we looked at everything. So for these cells, for the primary tumor to colonize the lung, the animal needs to have CD4 T cells and this and the animal, because they did them in separate experiments. They didn't do the conditional knockout like a crazy CD4 IL4. So they did on a CD4 last, and then they did on the IL4 knockout animal. And the IL4 knockout animal also do not make metastasis. So they concluded that you need CD4 T cells that would produce IL4 to like license the colonization of the lung. And this is all, it's very similar in the sense that it's all mediated by macrophages. You modulate the macrophage, then the macrophage produces factors that are growth factors for the epithelial tumors because it produces epithelial growth factor and then the tumors grow. So if this thing is like this, I have out, and then I'm gonna pass. But if you can read this, it's very good. Now it's just yeah, it's because I mean, no, it's like this. So if we have, oh, I'm going the other way around, right? So if like different organs, no, it's, oh my God, I'm sorry, people on the other side. If this thing is true, it's not very bad to think that T cells will modulate different organs differentially, and this will allow or not allow, or collaborate or not with the tumor. And then by this time, this paper from Polly Metzinger came out, and where she puts up like different tissues have different, let's say different, I like to say like different interleukins that modulate that tissues. This is not an exclusivity of the immune system. It's our physiology depends on those molecules. And the impact of different infections in different tissues would be different. This is this paper. It's a theoretical paper, okay? And then, but now we have ours. That's at the end. So just, I'm gonna try to go fast. So I asked Romulo Galvani, who is our bioinformatician by now, super bioinformatician, to check on the data bank if we have like if normal tissues could be separated just based on the RNA-seq. And so he found this and he said, oh, I can separate breast, lung, liver, and central nervous system. Okay, so let's take out, so he used a tool, the Excel, that has signature of immune cells and non-immune cells. Take out the immune cells. Take out the immune cells. It's still separated. Sorry, take out the non-immune cells. Let's stick only with the immune cells. It's still separated. Say, so probably we'll be able to separate that at the T cell level also. But then all they have in terms of signature for us to pull out which cells could be in there to separate the different populations, they don't have, they have just like gross phenotypes. So don't have like which cytokines each one is produced. So we started doing this, and just a preliminary data. I said, Marina is this super student, and then Felipe and Karina are also working on the same model, that get every antibody that we have for cytokine, and let's stain CD4 T cells from lymph nodes, spleen, lung, liver, and bone marrow, and see if they're different. Oh, and for that we do, this is important, we do the perfusion, but we also inject an anti-T cell antibody already labeled, so we can take out, eliminate the cells that were labeled intravascularly, in a way that we only analyze the cells that are inside the tissue itself. Okay, I'm gonna go to the easy one, this one. Whoops. Okay, so it was just like 4 animals. It's a starting point, but we publish now the idea of this thing on Frontiers in Immunology, a paper of theory and hypothesis, where we could add this data, because the data that I'm going to tell you what we are doing, it's going to take a while to come out. So we can separate so we do that, and then we do just like PC analysis, and we separate bone marrow just based on the 6 or 7 cytokines that we did for T cells, okay? Just based on that, we can separate bone marrow from liver and lung. That's really funny also. Lung, lymph nodes, and spleen grouped together. After the tumor comes in, and see, by day 17, I still don't have tumor in the bone marrow, but it's already a mass. So the phenotypes change, and now I can separate lung, liver, lymph nodes, and spleen. And after everything is already colonized, then I have another pattern that is different from this first pattern, but I still keep them separated somehow. Okay, so this was very preliminary. And the other thing that we can do is, like, if I look now, the same organ in different days, I can see that I can separate the different days, showing that they change their phenotype even before the tumor kicks into that organ. So what we are doing now, and we already ran, and we are analyzing, it's going to be a while, it's single cell sequencing to be able to identify which antigens are being recognized in each tissue and which is the pattern or the package of cytokines or molecules these cells are producing. But it's going to be a while. It's a lot of data. It's going to be a while to finish the correction. And the dream thing is this. The lady with a breast tumor arrives, you get the tumor, you would phenotype the T cells that are inside the tumors, and you can predict which organs are going to be colonized. Since these are the things that kill the patient and not this, right? So that's the basic idea. So a lot of people working, had worked and are still working on this project. And I really need to thank Ana Carolina Monteiro, who's super into bone metabolism now, and hormone control of bone and T-cell interaction and these things. Marina and Romulo as our bioinformatician, and Marina, who is suffering with all the single cell experiments, and the money, and thank you, and I need to do some ads. Okay. First symposium on immunotherapy. I don't know if it's going to happen because it's going to be, it is going to be in Porto Alegre. And the idea is to start a society of immunotherapy there. We have the symposium for the field cancer rede. In November, the LAMIG is Latin American Mucosal Society of Immunology, is organizing a course that's going to be great. The program is really good. The person that is doing that it's going to be at FIOCRUZ in Brazil is Herbert, that was also from here, right? And save the date, our neuroimmunology, our Congress of the Society of Immunology is going to be in neuroimmunology at Ceará at the end of September. Okay, thank you.
Thank you very much. It was a very exciting presentation, very exciting data. We are going to open first to students here in the room and all the students that are watching us through the Zoom room. So do you have any?
Oh, Bruno, sorry, I need to leave. Just want to thank and congratulate Adri. Adri, I have a couple of questions and also a collaboration proposal. So, but I will reach you by phone, okay?
This is good.
All right. Thank you so much, guys. Have a wonderful week. Thank you.
Thank you.
All right. If people online have questions, please raise their hands. We do have one question here. Lucas Bolini.
as usual. And thank you for your talk. It was really, really interesting. My question, and I think this is probably a more theoretical question because, of course, the path you took is a bit different. But of course, I think it's quite clear that T cells help shape the pre-metastatic niche. And of course, the changes in the bone structure are quite obvious. But I was wondering, how do you think the hematopoietic niche is affected by these cells during the tumor growth and the metastasis process? How do you think the hematopoietic niche in the bone marrow is shaped in this process?
Actually, we started doing that, but now, right now, I don't have anyone to do it. If you want, you're welcome, because I really enjoy hematopoiesis. At the end of the disease, the hematopoiesis goes really bad. But I mean, we have many, many bones producing. So to impact on the hematopoiesis, you really need to have a lot of bones involved on the disease. So like on a small amount of time, it's not going to impact. But what I think your question is, I would love to see the niches. And the tumor comes here. What happens to the niche that was a hematopoietic niche here? It will migrate up or down, will disappear, and it's going to be compensated at a different site. We don't know. And we also don't know how to do this thing. I don't think it's feasible.
My curiosity is really not necessarily like the systematic hematopoiesis, but specifically on the niche, like whether this specific niche does not have hematopoiesis at all at the end of the disease, or whether it produces more neutrophils or more macrophages of a specific phenotype. But yeah.
Probably at the site where colonization happens, the hematopoietic niche will be destroyed. That's my guess, but it's a guess.
Adriana, thank you so much for your talk, for coming here. And it's beautiful to see that I was there when you were starting the project. So it's really beautiful to see it all together. I wonder if you have an idea if the specificity of the T cell is important in that case, if they need to be tumor specific, or if the T cells that there are a lot of mature T cells in the bone marrow, if they happen to be educated by microvesicles or something that the tumor might be secreting.
We are still working on these things. What do I know? Apparently, I mean, there's no antigen, there's no tumor cell in there, right? And I know that the T cell needs to see the antigen. So what is the antigen that this T cell is seeing inside the bone marrow? Possibility one, it's primed on the outside with tumor antigen. But then inside the bone marrow, it might not need the tumor antigen itself anymore. Could be something else. This is one possibility. Possibility two, the antigen needs to get there. And one thing that we know, and it's already published, we published that also, and this is cute. The dendritic cell, if you get a dendritic cell, get this dendritic cell to present antigen to the T cell, tumor antigen, to a T cell that was already pre-primed. This dendritic cell will turn into an osteoclast. But this osteoclast that came from this dendritic cell is able to keep simulating the T cell on the same loop. It produces tons of IL-23, tons, and then the response with IL-17 and runcligand. So this is one possibility, that the dendritic cell comes from the periphery, goes to the bone marrow carrying the antigen. And the other possibility is really microvesicles from the tumor itself. But What we wanted to do at first was to go into the dendritic cell thing, because this bone loss is very intense on a very short time. So I keep thinking, how come like the 3% of total cells in the bone marrow are going to be T cells? So let's say that the frequency of the cells that have been activated by the tumor and modulated to have this specific phenotype is going to be 0.01%. So the number is very little to cause all this thing. So we're looking at different loops that would be enhancing this thing. So we wanted to do like a dendritic cell that need to be color transfected with a Texas red, whatever, and then transfer it and see if we can see osteoclasts that are red, just like this dendritic cell. But then pandemics, stroke, you need to wait a little bit, but it's planned.
And does it matter if the tumor is triple negative, or how it responds to hormones?
All our tumors are triple negative. So, but one thing that I think it's important to mention, and it's something that bugs me, and now I have an undergraduate student starting to do that, is if I add a T cell that comes from the 41 and simulate it in vitro with the 67 proteins, is it going to change the phenotype or not? And that's why we also want to do the single cell to define the TCR that is being recognized, because it might be that the modulation is dependent on the antigen specificity and avidity and affinity, and not on something that is being secreted on the environment. So this is something that I hope we can find out now with the sequencing.
Yeah, sure. Well, actually, we do have another question here. Then I do mine.
Thank you for your presentation. It was really interesting. I just wondering about your human data. I would like to know if you have some information about the patients that have the metastasis. I'm sorry. The type of the lesion is the same in the same site, or you have some morphological difference between them?
In general, in breast cancer, in general, in breast cancer, the lesion is osteolytic. In prostate cancer, is osteoblastic. So probably it's another loop that probably produces tons of OPG and not rank ligand. But usually the lesions are on the same places, like on vertebra and iliac and head of the femur, but are on the trabecular bones. The primary is the tumor. Oh no, we are doing triple negatives. They are all the same. But I'm talking to people at the oncoclínicas to see if you can do a big study now, and I want to include everyone. I don't want to separate by molecular type, because if this is this, this is this. I think.
Right, Professor Basinski.
Thank you. Very nice talk, as usual. So I have two questions. The first one is related to the previous one, and is that if you have this pre-metastatic bone marrow bone lesion in humans, and if this exists, if this can work as a signal for sort of controlling. And the second one, in this last slide that you showed, the production of cytokines by different organs, if those are, the dots are single animals or single strains. And you have done different strains to see if there is a genetic control of this mechanism.
Okay, the second questions. Our tumor only grows on biopsy. The tumors that we have, I mean, available to use for B6s, for example, they do not go to the bone marrow. But Carol, Carol é incrível. She got a tumor that grows in B6. Originally, it doesn't grow to the bone marrow, but she's forcing it. So she already have a cell line that injects on the memory pad. It's going to the bone marrow. Then I'll be able to respond to your question. Which was the other one? That could be. That could be, because then the tumor will stay there, the stick in. That could be. The lesion. In terms of the lesion, what happens is that usually they don't look at this kind of thing. They look when there's symptom and then there's not a rastreamento, tracking. They don't track previously. I mean, there are some exams that you do every, I don't know how many years, but not focus on this kind of thing. And there's this funny thing. If you use, the woman arrives with the primary tumor, and then most of them, especially postmenopausal ones, you start already what they call the bone protection with an anti-RUNC ligand monoclonal antibody, the Prolia, or bifosphonados, Zelotas, and it doesn't have any impact on the overall survival or number of lesions. So I think this thing of the T cell is that it goes there like, you know, hand in hand. Here, it doesn't, it's not widespread. It's very cell-cell contact.
Right. So you have showed that with the B cells transferred from the animals that were exposed beforehand to the 67 NR tumors, that these cells are protective. So I think the first question is, can these be exploited as a therapy? Can you identify what the hell the 67 cells are doing? And that's it goes a little bit beyond that. What's the difference between the 67 and the 41?
So we just sequenced the cells. Actually, we sequenced 67, 41, 66. 66 have a different metastatic pattern. We have a fourth one. It's called 168. It has another metastatic pattern. So we just sequenced and we are analyzing, but we will have that soon. This is one thing. The B cells, we did, because so if these B cells are that good, let's inject the B cells at different time points. It only works before metastasis hit in. Because what I think that happens is that there are many memory tumors that inject in the animal. People inject on the blood flow, and then it goes direct, or inject direct in the heart or inside the bone. But it's not metastasis. Metastasis needs to get out from the primary because there are a lot of difficult things that this cell needs to put up with to be able to arrive to the next thing. And then when it arrives to the next thing, I think that very fast, it becomes independent. The cycle, you know, and that's why, like, I'm not saying that all bone metastases are dependent on T cells. In our model, it is, and I believe we can contribute with that. But I think after a certain point, it goes. It only needs this first step to sit down, you know, like to accommodate itself in the bone marrow, and then it will go.
Oh, it actually matches this being an effect on these pre-metastatic niche, right? But what happens with the other metastatic sites in these animals that were transferred with the B cells?
I think it happens the same thing when we take out the rank ligand. And this is funny. The liver doesn't change very much, the same with rank ligand, without rank ligand. Maybe a little delay, but it will get there. The lung is boosted. The lung is boosted. So it's like the regulation. And then what I think it is that we already are starting to study that also. The regulation of the lung macrophages by T cells is somehow inhibited by rank ligand. So when I take the rank ligand, it's like the macrophages probably go crazy and produce a lot of traffic signals for the, but I mean, it's what I think. But it's weird. It's really weird. And I'm super, I find it weird. No referee had ever asked anything like that.
Mateus, do you have a question?
No, sorry, my
That's okay. Do you have any further questions? Anyone besides Miriam? Now I'm teasing her. Or Bolini? Okay. Miriam has a follow-up question, and then I guess we can finish. Miriam?
Okay, I'll try to finish with only one more. But it was just, I wonder if you have tried, because of your interest in dendritic cells, differentiating them from bone marrow and loading them with 67NR or 41 lysed, as we used to do with B16, and then see if you do. You did. Oh, okay.
Because we were interested on the, by then we didn't have the 67 worked out, but we wanted to see. We only did with a 41 because we wanted to see the transformation on the pro-osteoclastogenesis thing. But it needs the T cell. Only the soup or the protein extract doesn't do anything. It's like a priming.
Does it work to activate the
No.
endogenous?
No. Need the T cell to get it.
Thank you very much. So I didn't keep my promise. I did ask one more. But I want to, if there is really nobody else, then I want to thank you very, very much, Adriana, for coming here and for accepting our invitation, for being a beautiful excuse for Marcelo to join us here presentially. And thank everybody for sticking with us and discussing science with us, and we will be back next week. So thank you very much. Bye bye, Diego. Thank you.
Obrigado, pessoal. Até mais. Tchau, tchau.